So our first guest is Helena Telkänranta, and she has had a PhD in applied animal behavior science and work as a researcher on animal behavior cognition and welfare at the University of Helsinki in Finland, but also at the University of Bristol. Most of her research has been pig research, pig welfare on thermal image for several species, but she's also the founder and CEO of Arador Innovations, a research and science communication company where she works at the present time. Our next presenter today is Liza Moscovice. And Liza is has a PhD in biological psychology and has conducted postdoctoral field research with a diverse range of animal species, but her research integrates behavioral and psychological measures to investigate the role of social relationships and pro-social behavior in animals. She will give you more insight of that a bit later. But since 2019, she has been based at the Research Institute of Farm Animal Biology, which is mainly known as FBN in Germany, where she focuses on the social and emotional lives of pigs. I will now pass the floor to Liza, who will give you the introduction of the webinar. And please make sure that we pronounce your name correctly. And I give you the floor, Liza.
Thanks a lot, Gina, and thank you for all your work and efforts to publicize and facilitate today. I see there's still a lot of people entering. Well, you'll start admitting them. I've been doing it, but I hand that over to you for now. Great. So, and thank you for introducing us. As I think many people know now, this is this webinar is organized through PIGWEB, which is an EU Horizon 2020 project, with the goals to really create a community of pig researchers and also pig research infrastructures, where people are working together to harmonize protocols and best practices to promote high standards of pig research and also pig welfare. And a really important part of PIGWEB is, of course, also training and dissemination. So PIGWEB facilitates many different opportunities for training and also a lot of webinars, and that's how Helena and I ended up here. And we've actually met through PIGWEB working on a project together, and it's been really fun working with Helena, and I'm really looking forward to also giving this webinar and interacting with a lot of you. It's great that so many people enrolled, at least, and for us that really also shows how much interest there is in non-invasive physiological indicators in animals, and so that's really exciting to see. And today what we're going to do is first give you an introduction to basically what we mean when we're talking about physiological measures and why we think they're really important for understanding more about animal behavior and also improving welfare. And then we'll go more into the current status and also future prospects, kind of state of the art for non-invasive measurements of surface temperature, which Helena will cover in part 1. And after that, there will be time for question and answer and also a pause. So there'll be time there to interact and to take a break. And then I will cover the non-invasive measurement of hormones in the second part. And after that, there'll be another question and answer and kind of wrap up. So there'll be dedicated time to ask questions. We ask that you save your questions until those times. Of course, it's great if you want to write questions in the chat ahead of time. That's fine. And then we can also have those ready to answer at those times. We ask that everyone stays muted until the question and answer time. So just make sure you're on mute. And we also wanted to say that we will provide PDFs of these slides after the talk today. So don't feel that you have to scribble a lot of notes, or you can really just hopefully enjoy the presentation, take it in, and you're going to get a PDF of the slides for if that's useful for you after. Okay, did I say everything? I think that those was the main introduction part. So let's move into the background on why we think physiological measures are so important and how they link to behavior and welfare. And when we're talking about physiology, we're really talking about the study of the integrated functions of the body and the brain. And these functions are really important for maintaining homeostasis in animals. And homeostasis is basically the animal's ability to maintain an internal equilibrium in spite of, and in fact, in face of constant internal or external changes that the animal is faced with. And we can think of these changes or challenges that the animal is faced with in sense of physical challenges, such as changes in temperature that require thermoregulation, like this pig is doing with the mud rolling here. Also threats from predators that require mobilization of resources for a fight or flight response, but they can also be psychological challenges. And when animals are faced with challenges, they then go into a state of allostasis. And this is the time period, the processes by which physiological parameters are being modified, regulated to respond to these challenges, and eventually to return the animal to homeostasis. That's the goal. And so the physiological measures that we're interested in are really important for maintaining homeostasis, responding appropriately to threats. And we want to point out, that's what I didn't mention. Helena and I worked together on these intro slides, so this is really collaborative, and we were both talking about the most important points that we wanted to mention. And one of them is that I think when people think about homeostasis, they often think about it peripherally, what's happening in the body, but it's also critical in the brain, centrally. So just as the body needs the right balance of nutrients and hormones, the brain needs the right balance of neurotransmitters and hormones. And being in homeostasis or physiological equilibrium contributes to positive emotions, while imbalances in homeostasis contribute to negative emotions. So when the brain and the body are in homeostasis, the animal is more likely to be able to experience positive emotions. And when the brain and the body are out of this homeostasis, then this contributes to negative emotional states. Also, I think homeostasis is often, we know about these processes in humans, but this is true for all vertebrates, from fish to other mammals, to mammals. All vertebrates have similar brain chemistry needs and also similar origins of the experience of emotions that originate in some of the most evolutionary ancient parts of the brain, the limbic system, which is shown here in orange on this figure in humans and in rodents. So to understand homeostasis and what's happening when animals are in homeostasis, it's important to also have ways to assess emotions. And of course, emotions are very complex responses that involve correlated changes in behavior, in physiology, and cognition. But we can simplify emotions to try to find ways to actually quantify emotional reactions. And one way to do this that many of you are probably familiar with is the two-dimensional framework, where we think about emotions of having a valence component here from left to right, negative to positive on the graph, and also having an arousal component from low to high, and shown here on the axes. And the arousal component is mediated by the autonomic nervous system. Specifically, the sympathetic branch is triggering high arousal responses, and the parasympathetic branch is mediating the lower arousal responses. And of course, you could add on further dimensions to this graph. This is really just the beginning. So you could think about a third dimension of intensity, and that would be how strong the emotion is experienced by the animal. And we also wanted to point out that although intensity is sometimes correlated with arousal, it's not always the case. So a good example of a low arousal, highly intense emotion would be depression, shown here in the lower left figure, where we have very low arousal, negative emotional state, in contrast with something like an expectation of a positive outcome, something rewarding, which could be exciting, could be high arousal, positive emotion. So in the early days of animal welfare science, the focus was really on measuring negative states, negative emotional states, such as fear and pain. And the idea was that if we minimize fear and pain, we can reduce animal suffering. But in modern animal welfare science, we generally recognize that the absence of fear or pain is not enough to ensure that animals are not suffering. And so, as we mentioned, the brain also needs sufficient positive emotions to facilitate positive welfare. And this has led to a shift in the emphasis on identifying bioindicators of positive emotional states that could help to describe and characterize positive welfare. And another thing that we want to distinguish and clarify is the difference between emotions, the experience in the moment, and mood, which is more experience over time. And of course, welfare is going to be based on accumulated experience over time. And so you can think of these emotions over time as accumulating and influencing mood. And if you have net positive emotional experiences, this will contribute to positive mood. It's important for maintaining homeostasis. And here we can start to think about what we mean by good welfare. Not just the absence of suffering, but rather actually the focus on net positive experiences. And the opposite, when you have net positive, negative experiences over time, those are accruing and they're going to contribute towards a negative mood, potentially poor homeostasis. And in the extremes, we might have allostatic load, which is basically where the system is overloaded and can't produce the appropriate responses to return to homeostasis. And this would be a poor welfare state. So in summary, what's necessary for good welfare? Enough positive emotions to maintain homeostasis and this restorative capacity or resiliency, we could say. And that negative emotions should be infrequent and relatively low intensity. And that's kind of, I would say, the more current definition for good welfare. And it's a much more holistic view of welfare. And now the challenge, now I think where we are a lot with the field of animal welfare science is trying to figure out how to characterize positive as well as negative emotions over the long term. So we're going to talk about how physiological measures, and especially non-invasive ones, can contribute to that. So first, to get everyone ready for focus on animal emotions and how we're going to measure emotions, we first want to ask you all to contribute to a subjective, entirely subjective assessment of how animals are feeling. We're going to show you 4 videos. They're all pigs, of course, because our focus now is on pigs, but you don't need to be a pig expert to have an opinion about these videos. The point is to try to look for clues to determine or make a prediction about what kind of emotional state these animals may be in. And we know it's subjective, and we're really happy for many people to contribute. So what you need to do now is either scan this QR code with your smartphone, or you can also go to www.menti.com and enter the code shown here. I'll just give time for people to do that. And then in case you're late or slow or your phone isn't working, it's okay. The code will still keep on providing the code and the QR code and the information on the additional slides where we're going to show you some videos. And we're going to show you 4 videos, 4 examples. And after each one, we'd just like you to enter some words that are describing how you think this animal is feeling. And we'll show the video twice. And then this is going to make a cool interactive word cloud, so we'll be able to see what everybody has entered. So, are we ready? Hopefully people are, that's all working. So let's go over to the, well, actually, let me show you the first video then. So if you could, somebody tell me, are they already seeing this screen now? If they scan the QR code, are people here? Yes, on their own phones or computers? Hopefully that's the case. So I'm about to show you a video and you can enter your responses here. Okay. Oh, yeah, maybe those are actual, oh, those are responses. Sorry, no. You're ahead of me. Watch the video first. Okay, so here's the video, but it's great you started entering already. This is a clip, and focus on the pigs on the left who are having an interaction, and I'll play the clip twice. I see people were really even ahead of me, so maybe you can change your answer if you need to. So focus on those 3 pigs on the left. And the video should be playing now. Everyone should be able to see that, and I'll play it one more time. I see responses coming in. Okay, and one more time now, just focusing on the pigs on the left. What's going on for those pigs? It's interesting. I already see a big range actually of responses, so that's interesting to see. And also tells us why this is so difficult to assess just by observation. Okay. A few more people are still responding, but we're up to a good number, so I think I'll already show you. Can you guys now see this word cloud coming in? Gina, is that working? Yeah, okay.
Yes, perfect.
So that's what's interesting. We see basically, and of course the size of the numbers is saying how many scores these different terms are getting. So we have relaxed probably as the most scored response, but it's very closely tied with bored, curious, and comfortable. And that's interesting. So we have some bored, potentially negative and positive emotions. We have social in there, not well, chilled, worried, really a whole range. Okay, that's very interesting. I would love to know how many of these are from pig researchers and how many from people who don't work with pigs, but that's a different question. Okay, let's move on now to the second video. So first I'm going to, so I'll move forward, but don't say anything yet. Now you're on the second screen, and now you're about to see the second video. So how is this animal feeling? This one might be harder to score. There's not much behavior to observe. I'm playing it again. Okay, and that's actually time running, but there's not much else is going to happen there. So I'm just giving people time to put in their responses. They're still coming in. We can already show what's happening here. I'm sure you won't be too influenced by other people's responses. So we have bored now as the most dominant. It was also in the other example as a score, but here it's really more dominant, but then also relaxed, sleepy, stuck, sad, dead. The animal is not dead, I promise you. The foot moved. If anyone's noticed that, the foot didn't move. Her foot moved. So no dead animals in these videos, but I see why people would think that. Okay, so resting, tired, sleeping, sad, frustrated, restricted, bored. Bored is another dominant emotion here for this one. Okay, we're going to move on to the third example. Whoops, I made a mistake. I hope that doesn't bother you guys. Hold on. Okay, now here's the third example, and hopefully you're still on that screen on the Mentimeter. And so how is this animal feeling? Focus on the back numbered animal, number 4. Okay, I'm gonna play the video one more time. Okay. And we have a lot of responses coming in. Here you can see the responses, I hope. And we have now kind of a different reaction. We have stressed as the dominant response here, also nervous, but also something that could be potentially positive, like curious, but mostly negative. I would say negatively valenced words such as frustrated, anxious, stressed. Okay, and one last example. How are these animals feeling? This video is from Mark Bagaria and colleagues at IRTA, so thank you, IRTA, for the video. Okay, and one more time. For everyone coming in late, if you go to the menti.com and enter that code or just scan the QR code, then you'll see the link to enter in how these animals are feeling. Whoops, I didn't mean to stop that, sorry. Okay. I'll show you the responses coming in here. Here we have also positive emotions dominating now. Playful, happy, curious. Does anyone have a fighting? So there's some negative, potentially negatively valenced ideas too. Normal behavior, okay. So basically playful, happy, curious, and dominating. Also excited, exploring. Great. So thank you everyone for participating in that. I hope it also was kind of interesting for people, especially if you don't work with pigs so much, to see. I mean, these are all videos from myself and my colleagues from our experiments, and they're all conditions that pigs are found in. So this is in a very conventional setting, but we have pigs socializing, and I would characterize those as very friendly, affiliative socializing in a very relaxed setting. And a lot of you characterize that. I don't think people used low arousal, but the words you were using were basically suggesting that. So relaxed, even bored, potentially. But then there's also potentially this positive valence part if these social interactions are enjoyable for the animals and if they're reinforcing, if they are rewarding interactions. And here we have also a very low arousal state. I think everyone would agree this is low arousal. Some people thought the animal was even dead, that low arousal. And this is a farrowing crate. So for anyone who hasn't seen how sows are typically handled in conventional settings, before a few days before birth, they are usually restricted in these farrowing crates, and it's only for a few days, and soon after the birth, they should be freed. But so everyone said this is a bored animal, this is a stuck animal, so low arousal, negative valence. It seemed that was pretty much agreed on. This was harder for some people to characterize. I understand why. It's more subtle behaviors. But this is from an experiment I did with helping behavior. So this animal is actually trapped in this small compartment and needs help to get back to the group. And this is a high arousal, negatively valent states. For anyone who noticed the jumping up against the side of the compartment, that's an escape behavior. So that was showing the animal's motivation to get out. And the animal was very aroused, moving a lot of high frequency grunting. So this we could characterize as high arousal negative valence. And then we had also high arousal positive valence state, where these are pigs who are normally in a conventional setting, but they're brought outside once a week and get to be in a more natural state where they can show a wider range of their natural behaviors. And you saw a lot of playful behavior, potentially also some fighting or dominance related aspects, a lot of using the environmental stimuli. And so this should be a moderate to high arousal, positively valent state. And a lot of you were characterizing those pigs as happy or playful or explorative. So the point is, of course, to move from subjective to more objective ways to measure the emotional states of these animals and to gain insights through that into longer term mood and the animal's ability to maintain homeostasis. And so we want to move from the subjective, these ideas or our predictions, to finding bioindicators that can help us to objectively quantify what kinds of experiences these animals are having. And to do that, we can, sorry, it was frozen. So we can think about behavioral indicators. These are really critical, and hopefully everyone who's interested in physiology is either also doing behavioral research or collaborating with people doing behavioral research. So having an ethogram, a set, a clear description of behaviors that you're interested in looking at to characterize animals in your specific experiment is critical. Looking for things like changes in activity, approach avoidance behavior, vocalizations. There's a lot of work now with facial expressions in some species, not so much yet in pigs, but with tail posture and tail movement, there's a lot of work with pigs to try to see how these link to different emotional states. And of course, physiological indicators like heart rate, the blood flow to the skin, and hormones. And what's nice now is that a lot of these physiological indicators that before we could only measure invasively, we can now measure these indicators non-invasively, which is obviously critical if we're interested in observing animals without interrupting their behavior in their ongoing behavior and figuring out what that behavior, how it's important to them. So that is the introduction. And now we're going to move on to part one, and I'm going to turn the screen over in a second to Helena, who's going to give you the current status and future prospects for measuring surface temperature non-invasively. And after that, there'll be a question and answer session, and you can already start writing questions in the chat if you would like. So let me stop sharing. And now, Helena, you should be able to share.
Just a second, I will share there. Okay, good afternoon to everyone. It's really nice to be with you, all of you here online today. And thanks to Gina as well for all your organization and practical arrangements to make this possible, as well as thanks to Liza for all the great collaboration, including when we were planning this webinar for all of you. Now for the next about an hour, what we are going to do is that we will take a look at one of the new emerging, developing technologies and methodologies on how to use these animal-friendly, non-invasive remote technologies to measure the physiological processes that can tell us quite a lot about the animal's emotions, namely infrared thermography, also known as thermal imaging, as a way to measure surface temperatures of the animals, which again tell us a lot about what's going on under the surface. The image you can see here is a thermal image. Many of you have probably seen thermal images in the media or on TV or somewhere else. Maybe some of you have already used thermal cameras. Probably most of the thermal images that you have seen are these colorful, nice things. People even do art out of those nowadays. This is one type of thermal image, one way of presenting the temperature data in the image. What you usually see in scientific research is something like this, where we are utilizing the temperature data that has been stored in the form numbers in the thermal image. So the picture is just kind of a surface tool for us to get to the actual scientific data. I will start by telling you a little bit about the thermal cameras and this technology as a whole. Excuse me. So that if we first take a look at what are the strengths and what are the weaknesses or limitations of the technology that we are using in these measurements, then that will give us a better handle to look at what we actually can do and what kind of new research avenues we can explore with this technology when we are familiar with both its strengths and weaknesses. One of the hallmarks of a new field is that it has many names. You may have heard the name infrared thermography. It is often abbreviated as IRT, especially among researchers. It's also called thermal imaging, and sometimes you see other forms like thermo-imaging or something like that. They all mean the same thing, using a thermal camera to measure the distribution of a surface temperature of any surface, animal's face, animal's eye, landscape, houses, and so on. Some of you probably have seen thermal cameras in some of their many other uses. Thermal cameras are being used, for example, when looking for people who have been lost in forest or in the landscape. So then all you need for the thermal camera is to be able to detect this human-shaped warm figure in the middle of the colder landscape. Or then they are being used, for example, to identify in cold climates that if houses are leaking warmth from the house to the external world, and then you take a thermal image of the house from outdoors and see which parts of it look warmer. Like if the windows are warm, you can see that heat is leaking from there. And because there are many different ways of using thermal cameras, there are also very different types of thermal cameras for different purposes. Some are cheaper, some are more expensive. Like the one you can see on the left here is a typical model used by veterinarians and also customs officials on airports. Then the others are more commonly seen especially the one on the top is commonly seen in scientific research. The one on the right as well, the one on the top is mounted on a tripod. The others are handheld, but can also be mounted. The prices vary greatly from a few thousands to tens of thousands, and what they can do also varies greatly. If we are thinking of getting a thermal camera for research purposes, we need something that can give us those all-important numbers. We need a thermal camera that is capable of storing so-called radiometric data, which means that in each pixel in a thermal image, it is storing the temperature value of the surface in that pixel. And if we are working with animals, quite often the animals are moving, and so life becomes much more easy if you are not only taking single images, but instead you get a thermal camera that is capable of recording thermal video. which is just a sequence of individual thermal images. For example, if you have a 30 frames per second thermal video camera, then it gives you 30 thermal images each second, and then you can afterwards select the ones where the animal is in the right position for your research purposes. This is quite an important first step, because it is quite often I get approached by people, research groups, who have got hold of some kind of thermal camera and then decided to do a study, and then they end up with data that they can't actually make any sense of, and they ask then help of me or somebody else, and then they find out that the type of thermal camera was wrong for that purpose, and also there are many other things that have to be taken into account in planning to get successful research data, some of which we will very soon look at. So in addition to having the right type of thermal camera, even more important for research purposes is to make sure that the way we are using it is rigorous enough, something that can be repeated. A very necessary detail, a very useful tool is to write down a very detailed written protocol on how to carry out the thermal measurements themselves and how to carry out the data collection from the thermal images. And then when you have that rigorous written protocol, then that can be tested for interoperable reliability. Then you can have two different people independent of each other following the same measurement protocol and seeing if they come up with same or almost identical numerical data from there. For example, this thermal image is from a project that we were working on when I was working as a researcher at the University of Bristol in the UK. We were collaborating with the Institute of Animal Science in Prague in the Czech Republic. And this is one of the piglets in our study. And here we are developing the testing which way to best measure various parts of the snout and forehead to measure changes in the temperature at the tip of the snout and various different parts of the face that then can tell you about the what is going on in the autonomic nervous system, of which we will soon talk a little bit more. That's one of the most researched, the most studied avenues to use thermal images to see what is going on in the emotions of the animals. But the thermal imaging as such only takes quite a short time. Most of the time in a research project goes before and after. Before in developing the rigorous enough protocols for precisely that purpose, and then afterwards in the data analysis. So let's take a look first at how this technology works and what are its limitations, its weaknesses, because only when we know its weaknesses we can really tap the full potential of its strengths. So the thermal images, they are just a visual representation of the actual thing, which is the huge array of surface temperature data. For example, these thermal images, these are actually two versions of the exactly same thermal image, have been taken with a thermal camera with a resolution of 640 times 480 pixels. That means that there is that number of pixels, and in each pixel the thermal camera is storing the temperature value that it has measured from that very specific small point of the area that it is seeing. And you might be wondering why I'm saying that these are two versions of the exactly same thermal image, because they obviously look quite different. You can look at the scale on the right-hand side of each image. The scale is showing you firstly that how different temperatures are coded as different colors. So the image here on the left is showing in the grayscale mode, so that the lighter a pixel is, the warmer was the surface temperature in that place, and anything that is above 30 degrees will still be white, and anything that is below 15 degrees will still be black in this picture. Same thermal image, but now just with a different scale. Here you can see that here are 10 different colors that are being used to show the different temperatures here across the image. And so that the black, everything that is less than 1 degree will be black here. So you can see that also the bracket of temperatures that are being shown has been stretched, so that everything that is colder than 1 degree Celsius here is black, and everything that is warmer than 30 degrees is white. And then all the other temperatures have been coded with colors here in between. Here is another way of looking at the same thing, the same resolution of a camera. Here is this little rat, and I'm going to show you this picture several times so that it's the same picture, but we are just changing how to look at it afterwards in the analytics software. So for example, here you can see the grayscale mode, which is quite often used in research, because there you can see, even with the naked eye, you can see very fine detail in the gradients when something is slightly warmer or slightly cooler or something else. Now we change in the analytics software, we change it to show a little bit, no, at first we take some temperature data out of there. We can take a spot here from the tail, and the surface temperature there is 36.6. We can draw a line, for example, here on the forehead and nose, and it gives us the warmest and coldest pixel, their temperatures, and the average across that area. But then we can look at, this is still the same image, and you can see it's still the same temperature range from 23.1 to 31 is being showing, but now there are just a couple of colors added to make it more clearly stand out which areas are warmer and which are cooler. Then there are color palettes where you have more colors. This is again the 10 color palette. Then you can see even more detail here. And this is the same 10 color palette, but now we have just changed the bracket, showing in the previous ones it was 23.1 degrees was the lowest temperature that was seen. Now it is 7.1, so everything that is below 7.1 would be black here. Now you can see a bit of the surroundings where the rat is. And then again, if we want to focus, now the bracket has been moved upwards. Everything that's above 35.5 degrees is black. White, and everything is below 29.5 is black. So this way we can, for example, look at detail. We can see that the eyes of the rat, like eyes of any animals, are not uniform in temperature. They have these temperature zones. This is actually something that is being already used in human medicine, in eye medicine, that certain diseases in the eye first manifest in an abnormal distribution of these different temperature zones in the eyes. But the data is still the same, so we can still see that even if we can't see the tail, if we measure it from there, or if we can't see the forehead, we still get the same temperature values. So these are only different ways of looking at the same thermal image. So the take-home message of these images was that whenever you see a thermal image anywhere, you can be almost 100% certain that not all the available information in the thermal image is visible to your eye. And it also shows that when doing research, it is important to not think about it as a photography and collect a series of images, but think about it as a thermometer or huge number of different thermometers, little thermometers side by side. And then what you're actually doing is measuring temperatures in specific areas. And a brief look, still a bit deeper. Whenever we're measuring something, we become more successful in measuring it if we make sure that we have a good grasp of understanding what it is that we are being measured. So what is temperature? Many of you might remember from high school physics or somewhere else that fundamentally temperature is the vibration of molecules. So the so-called absolute zero, the 273 degrees Celsius, is the coolest possible temperature, because that is the temperature when the molecules or atoms are absolutely still. And whenever they are vibrating, they are warmer than that. The more they are vibrating, the warmer they are. And a byproduct of this vibrating is that every once in a while a photon, one of these very small fundamental particles, is emitted from the vibrating molecule. And the more the molecule vibrates, so the more photons it is emitting In other words, the warmer the surface, the more photons are being emitted from it. And a thermal camera is a device for counting these photons. You might be wondering why I'm talking about photons, because usually you hear people talk about infrared cameras and infrared wavelengths, and that is because some of you may remember the wave-particle dualism from particle physics. So whenever these elemental fundamental particles are traveling through a medium such as through air, they are traveling both as discrete photons, but also as a wave with a certain wavelength. And so the higher energy there is, the more photons you have, the higher is the energy, the shorter is the wavelength, because you have this kind of crepe-like thing, lots of little waves packed in a high energy state. And here you can see that all of the radiation, electromagnetic radiation that there is in the universe, only very narrow band of it we can see as visible light. Those are the wavelengths or the intensities of photon flow that our eyes have evolved to see as light, or our eyes have evolved to detect, our brain to interpret as colors. And then when you have lower flow of photons, longer wavelength, then you get infrared radiation. And thermal cameras are measuring mainly the mid-range infrared around here. That is still quite a wide range of wavelengths as compared to the visible light. And the infrared wavelengths have some similarities and some differences to visible lights. The similarities allow us to portray this data in image form in the first place, but there are also important differences to bear in mind when planning thermal imaging studies. One of them is that glass is invisible, as is water and perspex. Infrared radiation, the wavelength of infrared radiation happens to be in the area that is stopped by the molecular structure of glass or water or perspex. So if your animals are behind a glass wall, then you need to have some way of getting the infrared camera inside there, or some other way to make sure that there is no glass between them. There is a specific type of glass made of germanium, expensive, but it is available, that can be seen through by a thermal camera. So if you have animals who definitely are behind a glass, and if your institute has plenty of money, so it is possible to purchase these germanium, so-called thermal windows, that type of glass, and then you can do thermal imaging through it. If you take a thermal camera closer to the animals, it's important to remember that the lens of a thermal camera has a coating, very delicate coating, that is easily damaged. So it's important to make sure that the animals can't touch the lens, because they can then unwittingly damage the coating. Thermal radiation also, because it is longer wavelength, it is more easily reflected as compared to visible light. Here you may wonder, what are these two rats here, the image, or maybe three if you look back here. This is actually one rat, and in an arena where there are glass walls on two sides. So these are just her mirror images. Warmth or infrared radiation mirror images reflected from those glass windows. So as you can see, you can't see the thermal camera what is behind of the glass. And the strength of thermal cameras is that you can use it from a distance. Ideally, in very many research settings, if you do the habituation and planning properly, the animals will not even notice that you are taking measurements of them. But the remoteness is also the weakness, because when the photons are traveling through large quantity, long distance of air, there are plenty of hazards that can snatch these little photons in the middle of the travel. They can bump into dust particles and stop. There will be water molecules in the air which make them stop. And also because the thermal camera is only counting the photons that are coming to its direction, so any parts of the animal that are turned away from it, even curved away, will look cooler, because part of the photons will be escaping. And then if you have a warm heating device nearby, or sunshine coming through a window, or other animals very nearby, then then those will be adding temperature, adding warmth to the surface, and then the thermal cover will see it as warmer. And so the key, what I like to, one of the first things when I'm teaching this to students is that I ask them to please try to forget that people are calling it a camera, and just to think about it as an instrument that is counting photons, because that makes it much easier to plan and think about it in a successful way. So here are just some of the external factors. I'm not going through all of these, not to bore you, but you will be getting both a recording of these lectures and also you will be getting the PDF form of all of the slides that you see in this webinar. So you can use this as one kind of a checklist, different things in the environment of an animal that will affect the temperature readings in the surface. And then there are also a number of things in the animal himself or herself that will be affecting the surface temperature. So if we, for example, are interested in measuring emotions and those physiological processes that have surface temperature effects that give us information of the animal's emotions, we have to remember that all of these factors are also affecting the animal's surface temperature at the same time. This might look daunting, but when you actually have a good grasp of all of this, it is entirely possible to plan and carry out very rigorous and precise scientific studies on animals and even some observational, scientific studies, both of observational and experimental, and so that you can either standardize these or then some of these can be measured if they can't be standardized. And one more thing before we go into the question that how this is being used at the moment in studying emotions in animals and what are the developments inside. The difference between skin temperature or peripheral temperature, which means the temperature of the surface of the animal. So how is it that different with the body temperature? Because it might be easy to think about that, okay, we are looking at an animal with a device that is gleaning information, collecting data on its surface temperature. So we might easily think that, okay, so that will be the body temperature of the animal. But the skin is the biggest organ that we have, and it's a very active thermoregulative organ. So when the ambient air is cooler than the body temperature of the animal, then the animal's body, on purpose, it lowers the skin temperature, directs blood flow away from there to conserve heat. And similarly, if it's too hot, if the animal is producing too much heat from its own metabolic processes and muscles, and it's starting to feel uncomfortably hot because the temperature, weather is hot, then it will start increasing its surface temperature by diverting more blood to the skin. And so this means, but still the core body temperature is typically lower. So for example, here, if you look at this cow here, all cows have helpfully this little beautiful hair wall on their forehead where there is a little patch of bare skin. So there you can actually get a genuine skin temperature, even in this hair-covered animal. And you can see in this thermal image that the warmest pixel in this circle, the skin temperature here in this patch of bare skin is 27.8 degrees Celsius. So if the body temperature of this cow would be 27.8 degrees, then she would be dead. And so it's always like always kind of a good way to remember it is that the skin temperature is quite a good way to measure air temperature, because the skin temperature correlates quite strongly with their air temperature, but less so with the core body temperature. The skin temperature does reflect the activity of the autonomic nervous system, which Liza already a little bit referred to that in the introduction. Autonomic nervous system is the part of our bodies that has three parts: the sympathetic enteric part that takes care of digestion, sympathetic part which raises our heart rate and respiratory rate and adrenaline flow in the blood when we get aroused or excited, both in positive and negative contexts, and then the parasympathetic, which then calms the body and brain down. And these are the things that have been subject to dozens of published studies on how to measure animal emotions, or at least this one component, this one dimension of animal emotions with infrared thermography. And here you can see this kind of an basic situation, what happens when you have these dynamics of the autonomic nervous system going on. When something exciting happens, whether you are happily excited or you are afraid or you are angry, in all of those cases, your sympathetic nervous system gets activated. You get adrenaline released to your blood flow. You get increased heart rate. You get increased increased respiratory rate, and so on. So your mind and body are preparing for action. Also, your mind is in this kind of very alert state. And one of the things that happens is that the adrenaline that is being released in your bloodstream, one of its functions is that it causes vasoconstriction, which means narrowing in the small blood vessels that we have all over in our surface, on our skin, and in the inner corners of the eyes of people and many species of animals. Those little blood vessels are also clearly visible very near the surface. And so the point of this from the perspective of the body and its functioning is that when you are directing this blood away from your skin, your surface, then you are getting more blood in your big skeletal muscles that you might need soon for action. And because you get less blood in your peripheral blood flow, the thermal cameras these days are really sensitive enough to see the temperature drop that takes place there. Here you can see kind of the idealized curve. The time flows from left to right, and the temperature, let's say the temperature of the inner corner of the eye, is the y-axis here. And so here you can see kind of the baseline situation, nothing special is happening. Then something exciting happens, or something frightening happens, or something painful happens. And then you get this rapid drop. Sorry, I have to go the previous one. You get this rapid drop in temperature. And for example, in large mammals, it can be anything from 0.5 degree to even 2 degrees, depending on how arousing the situation is, how strongly the sympathetic nervous system is getting activated. And then you get this gradual increase back to baseline. In some situations, the temperature rises above baseline, especially in high intensity situations. And in others, it comes only back to the baseline. The reason for the drop is already known. It has been validated with physiological studies with animals with catheters, with cows with catheters in their bloodstream, measuring the SNS marker and other markers, other indicators in the bloodstream, and so it is. And it was found out that yes, the SNS marker, telling about the sympathetic nervous system getting activated, does neatly correlate with this drop in temperature. But then the rise in temperature, they were expecting it would be explained by cortisol. There was cortisol increase, but it did not follow the shape of this curve. So nowadays, the working hypothesis is that the rise of this is the sum of parasympathetic regulation, downregulating, calming down the sympathetic system, and again bringing the blood to the surface, and cortisol secretion, and possibly something else. Here's an example of one of the published studies. You can see it's Mary Stewart and colleagues from 2008. And there will be a list of detailed references later in this in the PDF you will be getting. And this was one of the seminal studies showing how it works. So here again is time going from left to right, the temperatures from on the y-axis, and then you have this. Here is the stimulus, which in this case, in this study, they were observing the temperature changes in calves that are being castrated in 4 different methods. So 2 of them were just kind of sham procedures. The calf was getting local anesthesia and not castrated, but just handled, and calf was getting not getting local anesthesia and just handled, and then 2 of them were actually castrated. One was castrated with local anesthesia, and one, horrible enough, there are some countries where you still can castrate animals without local anesthesia. You can probably easily guess which ones of these were the calves castrated without local anesthesia. They were the ones whose temperature in the inner corner of the eye dropped from 37 to 4 to 36.7. So approximately a bit less than 1 degree drop in about 1 minute of time, and then going above the baseline. So here you can see also that the ones who were actually castrated, their temperature rises above the baseline, and the ones who were not castrated, their temperature just returns to the baseline. Here in the image, this is also from their paper, you can see where they took their temperature data. So this is a calf eye, the calf. This is a profile of the calf. Calf is looking towards the left. And here you can see the place where they took the warmest pixel. So that is the caruncle, the inner corner of the eye, where there's plenty of small blood vessels. And similar studies showing that the temperature in peripheral areas, where there are lots of these narrow blood vessels, gets this rapid drop during emotional arousal, and then the gradual increase during the recovery phase, have been done in several species and several situations, and also looking at a few different body parts. So the animals that have been studied so far, they include mostly mammals, primates and ungulates and rodents and dogs and so on, but also some birds, chickens and blue tits have been studied in this way. And the body parts, inner corners of the eyes are very, very useful in animals who do have those little blood vessels visible there. Then also, most mammal species have vasoconstrictive ears. That means that the blood vessel structure in the ears allows them to constrict, to bring down the blood circulation on the surface. And all of mammals have vasoconstrictive noses. And so, for example, noses of different species of apes and monkeys have been studied a lot. Humans as well. In humans, it works exactly the same way. And in chickens, the combs and wattles, the fleshy appendages are very, very useful in this sense. You can see lots of temperature changes there. Most of the studies have been done in the setting of negative emotions. I think this probably reflects also the sad historical traditions in research. It's much easier to elicit strong and uniform arousal responses in animals by using some negative stimuli, such as frightening stimuli. When you use positive stimuli, you get a bit more diverse responses because there is more space when you are not in an emergency, but you are at an opportunity. Then there is more space for personality differences and individual experiences to come to play and bring little diversities on the nuances. But it can be studied, and it has been studied in positive contexts as well. For example, the Moe et al. Paper mentioned here, Norwegian researchers were looking at the same response, same drop and gradual increase in the combs of chickens when they were anticipating favorite food. And this, for those of you who are interested in looking in more detail into these studies, here is a list of some references that you will be maybe interested in. You will get this as part of your PDF. And in those papers, you will also find references to some other papers. So there are probably approximately 30, at least, well, a few dozen papers published on this way of monitoring or measuring animal emotions so far. So then a few words on how to become a successful researcher or any other successful person to use thermal cameras in whatever field you are interested in using them. And these are a couple of bit of boring looking slides because they are just checklists. But if you are interested in this kind of a research, you might find the checklists that you will have in your PDFs to be quite useful. And we are in the process, Liza and I and others are in the process also in writing a book. It will be on best practices, standard operating procedures, SOPs, for various physiological measures. So I will be writing the chapter on thermal imaging there. So once the book is published, there will be a much more detailed checklist. But if we look at this checklist here, so thermal imaging is, in a way, it is deceptively simple, because you can take a thermal camera and point it at an animal and focus the image and push the button and get a very beautiful picture. And you can actually do some very artful images using that. Back when I was still working at Bristol, there was this competition, art for science. The university arranged this competition that to people to submit pictures that are science and art at the same time. So for fun, I just sent them a little thermal video, which actually won the public vote in that contest. So you can make beautiful art very easily, just point an animal with a thermal camera. But if you want to do groundbreaking research, then a really good understanding on how all of it works is crucial, and will be your superpower in this kind of research. But not only, we talked a bit about understanding the thermal camera, there's more that you can understand about them to become successful, and also to understand the animal that you are measuring. The physiological processes that you are going to measure, and how those physiological processes are connected to the myriad interactions in the brain and body that are involved in an emotional interaction. The better you understand the physiology of the animal and how those physiological processes function over time, the better you will be in planning that, for example, if you have an experiment where you are presenting some stimulus that the animals will be reacting to, then you will know how long before that stimulus you will have to start recording, and even before that, in which way you have to, on the previous days and possibly weeks, what to do to make the animal genuinely habituated to the presence of the thermal camera, so that will not affect. And then you will know that how, when you have a good understanding of the physiological process you are measuring, you will know that how frequent you will need to sample. Do you need a continuous video or samples, for example, every 30 seconds, or what, and for how long you will need to continue. For some of the recovery processes from the graph I showed you, it may take, right now we are analyzing the data set, where one of the questions was that what could be kind of the longest recovery times after positive arousal excitement in a positive context, and it seems that half an hour definitely is too short. We were looking at them for 2 hours, and it might be that something a bit more than 1 hour in that context and those species is enough. But so when you understand the physiological processes, you will plan your experiment successfully. Also, it's important to understand the other physiological processes. For example, physical exercise has very similar surface temperature effects because it has similar effects on the autonomic nervous system as emotional arousal. And if the environment is too hot or too cold, that also has some similarities. So when you know those well, you will be successful in planning your experiments. And then, of course, the mechanisms through which these affect surface temperature, so the better you understand how, for example, the surface circulation works, the more successful you will be. For example, if you want to test new sites in the body, where else to measure in addition to inner corners of the eyes or noses or ears. And also anatomical knowledge of your species. It helps you also in determining where you can predict that these vasoconstrictive effects will be most visible. And then, of course, in any study on animal emotions, if you are using any measure, especially quite a novel measure that this still is, it is always useful to have other independent measures, either physiological measures and preferably also behavioral measures, such as video recording of behaviors, and then analyzing those videos in detail with some standard ethogram description of relevant behaviors afterwards. to be used as so-called ground truths, so something that will give you an external independent information of the animal's emotions. Furthermore, just to kind of reiterate what we mentioned at the beginning of the technology. So at first you need the right type of camera. It is nowadays quite common that various research institutes do have thermal cameras that may have been bought previously for some other purpose. Some of them will be suitable for emotion research and some of them won't. So to get familiar with the technological capabilities is useful or necessary. And similarly, you will need to know what kind of sampling frequency you will need, not only in terms of that if you're taking single images, how often do you need, but if you have moving animals and you take video, how fast do they have? For example, with pigs, 30 frames per second is usually enough. For chickens, it is, for many purposes, it is too slow. Chickens are very fast, especially in the head movements. And so but it also partly depends if you do have a camera with 30 frames per second, and if you want to do studies with chickens, one way of countering that is that if you plan it so that you will be measuring the chicken in profile, they usually if they keep an eye on you, they will move more slowly when you are on their side as compared to if you are right in front of them. Then they will have these more rapid head movements when they are alternating which eye they are using to look at you. And the resolution. So how sharp or how many pixels do you need thermal camera to have? And before you start the experiment, it's important to become familiar. There are two groups of people or beings that need to become familiar with the camera, yourself and the animals. So with yourself, the most important thing is to do a lot of practicing on how to focus the camera. So thermal cameras are focused in the same way. Most thermal cameras are focused in the same way as ordinary cameras. They have a ring that you rotate with your fingers until you get the image in perfect focus. And focusing is important because the data is much more accurate if you have a focused picture. If you have a picture where the animal is out of focus, then the picture will be blurred and the data are kind of bleeding. The pixels are kind of bleeding data to the adjacent pixels. And so you can do much more with the images if you make sure that you want to take sharp images, and practicing of focusing, especially with moving animals, takes some practice so that you get a good handle in your wrist movement and finger movement. And the animals depends on species and especially individual, and especially on how much strange equipment they have already seen before. So the time, how long you need to habituate varies, but the most important thing in habituating animals to this or anything else is that the criteria, when the animal is habituated enough, the animal itself should be the criterion. So not that you decide beforehand that you will be spending 3 days habituating the animals to the camera, but you decide beforehand that what are the behaviors in the animal that are reliably showing you that it is no longer paying attention to the presence of the camera. And it's also useful to habituate them not only camera that is not running, but also to a thermal camera that is running, because most thermal cameras, when they are started and when they are auto-calibrating itself every once in a while, and when they are turned off, they emit ultrasound, which many mammals are capable of hearing. So that's why it's important to make animals habituated also to a camera when it is already running. And then the emissivity that is mentioned here, that is, many of these are things that I could spend half an hour or more talking about each of them. So also emissivity is easy to spend half an hour talking about, but here just quickly, emissivity is a measure of, it is a number between 0 and 1, and it tells you that how much of the temperature radiation that is coming from the surface of the animal is likely to actually come from the animal as compared to not being reflected from the environment. So the smoother surface, the more reflected temperature there will be. Human skin has an emissivity of 0.98, and there have been some studies, including in pigs, finding out what actually are the real emissivity values for those types of skin. And thirdly, in this checklist, so whenever you are imaging, so then these routines that you quickly become familiar with, you measure with some external meter, you measure ambient temperature and humidity, and you measure or estimate the distance to the animal, and you set those in the camera before starting measuring. And then after you have your thermal videos, then it's time to, or maybe even before you ideally before you plan or when you plan the experiment, but at least after you have your thermal videos, then it's time to decide rigorous criteria on how you define the areas where to collect temperature data, which thermal images are sharp enough, and where the animal is in the right position, and then do some inter-observer reliability testing to make sure that your written protocol produces the same data or almost the same data with different people. So where are we now in the science? As you could see, the studies in emotion mostly involve the increased arousal. Some of them have been following the whole curve, the rapid drop and gradual increase in peripheral temperature. Some of them have only been measuring the rapid drop in the beginning. And some methods are at the moment, some approaches are being tested to find ways to measure valence, whether it's positive or negative, but that is still at the early stage. In physiology, the science is more advanced. So measuring thermoregulation, so how the animal's body interact with the ambient temperature, that's one of the most advanced ways of using thermal imaging in animals, and it can be used in many settings, like monitoring animals in zoos or on pasture or elsewhere to detect heat stress and thermal comfort. And in veterinary medicine, there are, for example, here the cat here, the ambient temperature is about, well, it was a hot day, and you can see how the cat's ears are shining white. So this is one example of thermoregulation. So the ears have turned on their cooling mechanism. They are channeling lots of blood near the surface to get rid of some of the body heat. An example of this physiological studies in thermoregulation. In veterinary medicine, there are also various applications already in use, like early detection of mastitis in cows and thromboembolism, so the pulmonary block in the blood clotting in cats and other uses. It might sound surprising that it's quite difficult to measure fever with thermal imaging, but that's because thermal cameras are measuring skin temperature only, and as you remember, skin is a very active thermoregulating organ, and as we know that at different stages of fever, the skin is actually conserving heat at some stages, so that the surface temperature is changing up and down, partly not in sync in what the core body temperature is, where the core body temperature is going. So there are when there is prolonged fever, there has been for some time, or at late stages of fever, then the surface temperature is warmer than normal. But it is not as easy as that. It's not so that whenever an animal has fever, always the skin would be warmer than normal, because the skin can be actively thermoregulating and cooling itself. And so, as you can see, this is still in terms of animal emotions and also animal welfare. Here, the title here says pig welfare, but this also applies to animal welfare in all the other species, because the research is approximately at the same level in various species. So there is no one single species that would be far more advanced in these studies than the others. And so if we think about emotions, how to measure emotions in terms of measuring welfare, to get from here to the future, if we think about if we think that the time is going from bottom to top in this slide, and if we have these three different things that we want to measure in terms of animal welfare, the veterinary applications, and the like detecting inflammation and fever can be also used in monitoring on farms and elsewhere. and thermoregulation, thermal comfort, and emotions. So at first, if we look at the building blocks when we go from bottom to top through time, there still is more to find out in which of the physiological processes that are linked to emotions do have measurable surface temperature effects, as well as how they affect with the other things that are going on, metabolism of food and the effects of exercise and so on. And then how different species are compared to each other. The underlying physiological mechanisms are very similar in both mammals and birds, all species that have been studied so far, but the anatomical differences, for example, faces, do produce quite great differences in which part of a face you actually do find the temperature changes that can be measured. And then there will be needed studies to look at independent measures, the behavioral measures and other physiological measures, heart rate monitoring and so on. And then we come to the point where we have better measures than we have now to use infrared thermography by scientists in control environments. And then we still have to look at a lot of environmental confounders like ambient temperatures and moving air and so on before we can get to practical measures that can be used on farms and so on. But this is one of the areas where the science is actually contributing quite a lot to the animal welfare research in general. And like in the beginning, in the intro, when Liza was telling you about the difference between emotions that are the short-term experiences here and now, and then the moods that are, it's kind of the cumulative sum over time, kind of the baseline situation that the animal is feeling when nothing special is happening. So already at the moment, there are methods to study not only emotions, but even moods. Judgment bias test, which measures whether animals are reacting in an optimistic or pessimistic way to a task that they have been trained for, and they're varied. And also other more species-specific behaviors. Heart rate variability is one quite nice physiological measure for long-term welfare and mood, as well as the dynamics of cortisol and DHEA. Cortisol alone, as Liza will soon tell you, is quite a complex thing. Well, of course, everything else is too, but there are ways that you can use cortisol and other things to get some information on long-term welfare. And then there are developments underway. For example, judgment bias tests are very slow to carry out because you need a training phase. Attention bias test is being tested whether it could be a quicker alternative. And then there are IRT measures, infrared thermography measures for further information that can be gleaned from extracted from the dynamics of the sympathetic and parasympathetic nervous system, and then the hormone measures that Liza will be soon telling you more about. And these IRT measures, the thermal imaging measures, are being developed in several places, several universities, several of which we also, me and my group are also collaborating with. And similarly, our research company where I work now, Arador Innovations in Finland, and its research arm, Arador Health Science, we are also developing new types of software for both researchers and veterinarians, and use to measure thermal imaging, to measure animal welfare and health with thermal imaging. Thus, that is still a work in progress. And for anyone who is interested in planning or using thermal imaging in their work, the education and practical skills are essential. And one opportunity for that would be that in Arador, we are arranging courses in thermal imaging next winter in January and in February in Finland. There will be 3 different courses. So in January, there will be one specifically for veterinary clinics. And then in February, there will be one for zoos. And then later in February, there will be one for scientists. And for each of those, we have tailored the content of the courses so that they will be as relevant as possible for these fields. But then anyone is welcome. So even if people are not working in these fields specifically, but would like to learn these skills, that is also very, very useful. And we have just opened the registration for these courses. You will find out more in the email address that you can see here. And we have now an early bird registration price. Actually, we have a super early bird discount, which is until the end of July. So if you register in July, you will be getting it much cheaper than the normal price. But on that website, you will be finding more information on that course. So that's one example of in-depth. It will be two days of lectures and practical exercises in the conference center, and then 1 day practical exercises actually with animals in real life facilities, and plenty of different types of thermal cameras to try out and practice with. But so that was basically what I had in mind to tell you today, and I think I have pretty much filled the time that was allocated. So I will turn my camera back on. I was keeping it off not to have my face disturbing you while talking, but you might have some questions on mind. Gina, will you be moderating the question session? Yeah. Okay. So thank you for all your attention, and let's proceed to what kind of questions you have in mind.
So we do have a first question, but thank you very much, Helena. We do have a first question in the chat from Neil. And he says, very insightful presentation and for the tips. The question is, if animals are dirty, or maybe there's a high presence of flies, would this significantly affect the results collected in temperature readings, especially when they get soiled with their own manure, example, pigs in pens located in intensive farms?
Yeah, that's a good question, and the short answer is yes. And it is right that, as you can see in many thermal images, there's actually even an example of that in this image of pigs. Do you still see my screen?
Yep.
Yes, yeah, and you see the picture of pigs here?
Yep.
Yep. So you can see, for example, that here is a darker dot. So that kind of a thing has to be some kind of a speck of dirt on the surface, and you can see that even a very small and thin little spot of dirt makes the thermal camera to believe that this specific part of the pig skin is clearly warmer, cooler than the other. Yeah, here in this picture, you can also, this is a picture of environmental confounders as well. These pigs are half in sunshine and half in shadow. But yes, so any type of dirt or manure or excuse me, pieces of straw or droplets of water, all of those will obscure the real temperature of the skin at that specific point. And one way, the most common way, and probably the most useful way to get around that is to measure such body parts that are not covered by dirt. The inner corner of the eye is very useful for this purpose, because even pigs who are very dirty, they still, fortunately, they normally do not get dirt inside of the eyes, so that the coronal of the eye, the inner corner of the eye, still remains clean. And so that just means that then the study has to be, the experimental setting has to be planned in a way that the thermal camera is low enough and positioned so that the inner corner of the eye is visible to the thermal camera. Yeah. Other question?
Feel free to just raise your hand, shout out questions, or write them in the chat. I see a question from Charlotte.
Yeah, hello, thank you for your presentation, very interesting.
Thank you.
Yeah, I'm considering using this technology. And I am specialized in laterality research, and I think you already published in this direction. I wanted to know your, like, to me it's really complicated to know the physiology behind it, because especially for facial like thermography, when for example, one ear or one eye is hotter than the other side. So usually the innervation is like contralateral, but so to me this is about vasoconstriction. And is it about like the brain that is getting more active on one side and then it's hotter, but at the same time there is the skull isolating it? So is it reflecting the contralateral brain that is more active, or is it reflecting? Sorry, I'm not sure if my question makes sense, but
that's a very relevant question, and as you probably have seen some of our publications on early publications on this. I didn't mention it here because the study of the whole thing is still such an early stage that we don't have any kind of ready-made methodology to provide yet. And the reason why it takes some more time to finalize the methodologies is exactly what you said, that there are actually several different lateralization processes going on at the same time. And a bit more clearly, several different physiological routes. There's only one lateralization process usually at the same time, but there are several different physiological routes that are affecting the surface temperature. For example, one study, at the moment we have, my group in Bristol, my PhD student there, Marco Ramirez, and others of us, we had just submitted to the animal welfare, we submitted a study in calves where we measured very many areas around the eyes, so the inner corner of the eye, the muscles around the eye in the inner side, the muscles around the eye on the outer side, etc., and found out that there are lateralization responses and that they are to different directions in different situations, and which again tells us that there are more than one physiological pathways through which the brain lateralization causes surface temperature effects. And our working hypothesis is that there are at least two processes that we can think of, one of which is direct heat convection through the tissues in the head from the brain hemisphere to the eye that is near there, because skulls are quite well insulated to heat, but helpfully evolution has provided skulls with holes for eyes, and all those tissues are quite wet. So when you think about the route of the tissues from one hemisphere of the brain through the eyeball, which is very wet to the surface of the eyeball, that's actually quite well conducing to heat. So it is theoretically entirely possible that when there is more activity in one hemisphere than the other, and we know that from brain imaging that there is then more blood flow in that hemisphere. So if the activated area is close to the frontal part of the brain, like in the frontal lobes, then you could theoretically expect that there is some heat convection that comes all the way to the eyeball. Whereas at the same time you have as you said, that the nerves, the ocular nerves are going contralaterally, so that they are kind of crossing. That your right eye is communicating with the left hemisphere, and your left eye is communicating with the right hemisphere. And actually, we did see that that the muscles around the eyes are functioning according to this logic, and so that when in situations where you would expect, for example, left hemisphere, in positive situations, you would expect that to activate, then the muscles on one side, I think it was the inner side, but not the outer side, one of them. So muscles around some part of the eye were the ones that were getting activated. And so we still have some work to do, further research to do, to make sure that we understand this correctly and that we can untangle this correctly to find out that which parts of the head are reliable enough indicators of which part of the brain being activated.
Thank you very much. And
thank you.
If I may continue asking, if we have time, did you consider combining this, for example, with FNIRS, functional near infrared spectroscopy, because then you have direct signal of the brain, and then to validate?
The idea has crossed my mind, but I have not yet contacted what's his name in Switzerland, who specializes in this, and Lawrence Gigax, I don't know how to pronounce his name, who was one of the names working with FNER. So I have been thinking of contacting him and asking whether he would think it would be. I think because in the development stage, FNERs would be one of the independent physiological measures that could provide some information on what is going on there.
Okay. And
Yeah.
sorry again, because you mentioned the eyes again. And I had in mind that, for example, the pupil of the eye is represent a hole and should be colder than, or like that it's completely different than the rest of the eye on the surface. So when we have a signal of the surface temperature of the eye, the pupil should be significantly different from the rest of the eye, but
yeah, the pupils are visible if in thermal images, if you have high enough resolution and you are close enough and you have sharp enough image, the pupils are visible. There is a temperature difference, but it is actually not that big. I think it partly is because it is still a liquid-filled cavity.
Okay.
Yeah, but we have been doing some looking into the internal anatomy of the eyeball, but not necessarily enough yet to be able to do very good predictions in that field. But you are right that it is different, and it is visibly different in thermal images that are taken close enough.
Yeah, because I heard from only infrared camera, like not infrared thermography, you can detect better pupils on these images because they are completely like different structure than the rest of the eye, and that's why I was asking. Yeah, but
that is true. Yeah.
Yeah.
Thank
we
you.
want to leave some time for other questions. I see them coming in. Thank you. So the next question from Laura is in the chat. Many IR cameras that have been used have a resolution of 320 by 240 pixels. So considering, for example, the lacrimal caruncle of the eye, which is such a small area, how does this pixel representation impact the accuracy and reliability of the study?
Yeah, that's a good question, and the answer is mainly that it depends on how close you can get to the animal, and partly also that what size of an animal you are studying, because, for example, if you are studying, let's say, horses or cows, then the lacrimal caruncle itself will be bigger than, for example, if you are studying rodents or rabbits. But the main difference is in between how close you can get to the animal. Because if you, for example, in one study we did in rats, hasn't yet been published, we did some further analysis, but we were imaging them from the distance of thermal camera that we had then was an older model. It was the resolution was only twice as high as yours, so in the same order of magnitude, and we were at the camera lens was at the distance of 70 centimeters from the rat, so we had a food rewarded food rewarded task that the rats came to do. In all of our studies in our group, we do either positive studies, or whenever we develop methods to do anything negative, then we are not causing any pain ourselves, but we collaborate with veterinarians and others who already have those patients. But anyway, so those rats, they were trained to come there and eat at the distance of 70 centimeters, and that camera that was quite similar to yours could pick up the lacrimal caruncle very reliably, even that size of an animal. So the main question is how close you can get to the animal. If you can get to the distance of a few meters from a horse or a cow or a pig, then that will be sufficient even with that type of a camera.
Oops, sorry, I'm muted. Great, thanks. I think we just have, I'm just gonna move it along so that we can have time for a break. I think we have time for one more question here, the follow-up question, which is, what's the clinical importance of minimum, maximum, and average temperature variations across different eye regions, especially for veterinary practice? So do you have recommendations for when to use minimum, maximum, average?
Yeah, good question. It depends entirely on what you are trying to diagnose or detect, or what is the question. For example, that if you are looking for abnormalities in the temperature variation across the eye, like if you remember the one of the slides where the rat was, the color scale was zoomed in, and the rat's eyes were showing these different zones of different temperatures. So for example, if you are looking for abnormalities in that, then you would need to look at, for example, 4 or 5 different parts of the eye with the knowledge of what would be the normal temperature difference between them, and then look at each of them. And so for that kind of question, I would recommend you to take a small area. A minimum of 9 pixels is usually recommended, can be bigger, from each of those 4 or 5 points across the eye. And so measure all of these areas separately for each of them, get the average from that circle, from that area, and then compare those to each other. The minimum, maximum, and average are just they only mean it's just from the analysis software that works in a way that first you denote an area, you draw a line, or you draw a circle or a rectangle or some shape, and then the software gives you the answer that what is the average temperature over this entire area you marked, and what is the coldest pixel here, and what is the warmest pixel here. And for almost all purposes, what you actually need is the average temperature across the area that you are looking at. But for example, one of the few exceptions is that if you are looking for the carbuncle of the eye and want to know its temperature, then helpfully the carbuncle is the warmest point in the entire eye, the point with the warmest surface in the entire eye. So then you can draw a circle around the entire eye and ask what is the warmest point here, and just make sure that it is in the carbuncle area. And in that case, you would use the maximum value that the software gives you as the warmest pixel in the carbuncle. But for most cases, you just draw the area that is, or for interest, you make it small enough to make sure that it only includes the area that you're interested in, and then you take the average temperature of it. In animal studies, you very seldom need the minimum temperature, because quite often the minimum temperature, the coldest pixel in your image, quite often is a speck of dust or drop of water or something, some confounder on the surface of the animal. The only time I have needed myself have needed the minimum number in research has been when we were looking at eye temperatures in chickens, because in their eyeball is cooler than the surrounding skin, and in that case that helps you find the standard point in the eye. But in short, for most purposes, if you want to know the temperature in a certain area, you just define the area with the analysis tool, and then ask the analysis tool the average temperature of that area.
Great, thanks a lot, Helena. And I see thumbs up on all of those questions. So it looks like people are happy with they got their question answered. Let's move on now. So I see that I want to have time. Also at the end, there'll be time for more questions for me, for my section, and also for Helena, of course. So let's take a 5-minute break so everyone can stretch and have a pause. And at 14:50 exactly, we'll start the next part on the focus on hormones, and then we'll have more time for questions at the end. So see you in 5 minutes, everyone.